So if everybody could go to their fast data directory and run this command, and that will copy across the files that you'll need for this practical session. And that should be pretty clear to everybody by now, exactly what that line of code is doing. It's copying recursively everything in that directory to our current location. Does everybody have those files copied to their local area now? No? Pardon? Yeah, you don't need the PDF file. What you need are the two files. The ones that you particularly need are the bug binary and bug snips. And if you go back to the Google Drive directory where all the other things are, the PDF is actually in that folder now as well, so it's much easier for you rather than having to try and get it out of terminal. Huh? Okay, hang on a second. Let me just... These are the two files that you particularly need. And if they're in a folder called Gemma example, it will be really good because then you'll be able to just type exactly what's on the screen. The PDF file is in the Google Drive that you have access to if you go back out where you got all the other PDFs from and refresh that page. It's there. If you don't refresh the page, you won't be able to find it. Okay? Yeah? Somebody will come and help you. Anyway, in the last day or so, we've worked pretty hard on the mechanics of actually dealing with sequence data and getting it into a state where you can start to do some biological questioning with. And we heard from Stephen first thing yesterday morning. How do I make this full page?
Somebody will help you.
Who knows how to use this operating system? Yeah, I want this to be a slideshow. Instead of a instead of a sideshow.
The menu will be shown if you put the view. Is the
full screen.
Remove the continuous because otherwise it will be continuous, and then just full screen or F11. And then if you want to remove that, you need to go out and close this before going full screen. Now and to go full screen with F.
Okay.
And to pass, I think that is
okay. So We learnt yesterday about how, from Stephen, about how we're trying to connect the genome to the phenotype. And that's basically what GWAS do. We try and figure out what's going on inside an animal that creates a particular phenotype. And Stephen used a particular example of a giraffe. I apologize if you're easily offended. But you know, as the environment changes, we might see changes within an organism that are reflected in their phenotype. And we're trying to track exactly how that's happening. And we use GWAS to do that. So the whole concept of a GWAS is to evaluate the association between any genotype marker and a phenotype of interest, whether it be a long neck in a giraffe, or stripes on a tamima, or body size, or anything in Mytilus, or whatever phenotype you like. Okay? You score that across a large number of individuals, and we try and figure out what genetic associations are there. So the way I'm going to talk to you about that today is using a program called GEMMA. And I think I have a little bit more kind of theory for you first. Yeah, press space. Okay. So there are some problems with association scans, and that is that you need a large number of individuals. And so the power to detect an effect is related to the number of individuals that you sample and the strength of that effect. So if you have very small effects or multigenic traits, it's very unlikely you're going to be able to find them using GWAS. And here's just an example where we have the effect size on the y-axis, and as sample size increases, we get a better chance of actually finding those effects. But if the effects are small, we're not going to find them. I'm actually going to ignore that other side of this panel. Okay, so within any population of genotype markers, we have actually several populations of SNPs. Okay? We're going to have a whole bunch of SNPs that actually have no association to the phenotype. We're going to have some SNPs that have small effects on the phenotype. We're going to have another population of SNPs that have large effects. And that's depicted here by these three, in this case, different shaped curves. Okay, and what we want to do is try and explain the amount of the variance in a phenotype that is explained by the SNPs that have an effect and any other random effects, which is all those other SNPs that have no effect. And we want to explain the amount that is just associated with those SNPs of large effect. So GEMMA stands for Genome-wide Efficient Mixed Model Association. Are you guys ready for some maths? Yeah, everybody's coffeeed up. Okay, so we can run linear models, we can run linear mixed models, we can run multivariate models, and we can run Bayesian linear mixed models. I recommend, if you're going to use GEMMA, which you all are because it's a really good program, to jump into Google, not now, but in your own time, and just Google GEMMA manual. Make sure you put manual on the end, because if you just Google GEMMA, there's a very pretty model that you'll get lots of pictures of. And download that PDF, or download pictures of the model if you like. And it contains a very good walkthrough. So, some maths. So the top equation is the linear model. Right, so who can tell me what this second term is? Oh, come on, I'm joking. All right, so the problem with the linear model is that it assumes that all the SNPs come from the same population, and that population is normally distributed. In that there are some SNPs that have a really high effect and have really big effects. There are some SNPs that have low effects, and that the bulk of the SNPs have some kind of intermediate effect. And we know that that's not true. Right? Linear mixed models are a little bit better, because they say, okay, there's a population of SNPs that has no association with the phenotype, and there's a population of SNPs that does have an association with the phenotype. But they're limited in that all of those models assume equal variance in those two populations. And again, we know that that simply isn't true, because if SNPs have no effect, then there's no variance around that having no association. So we have to have a model that will allow us to deal with 2 populations of SNPs, or more, 3, 5, 7 different populations, all overlaid against each other, that are allowed to have varying degrees of variance in each of those subpopulations. Everybody with me still? Yeah? No? Who just wants to go home? Okay. So the Bayesian sparse linear model is a Bayesian process, so we're not actually dealing with exact things. We're approximating our phenotypes, our genotypes. And GEMMA runs on 2 different things. Has everybody heard of Plink? Hands up if you have heard of Plink. It's a file format, it's based on pedigrees. No? Okay, good, because I'm not going to talk about that, but you could use Plink files if you want. Okay, so we'll just grey that out. So what we're going to use is BIM-BAM genotype format. And Victor has written a script for us. It's BCF to Geno. Now, of course, we mapped all our reads onto our de novo assembly, and then we called SNPs overnight. And that's why we started up first thing this morning, and here we are, because we called our SNPs last night. Okay, we didn't. But I've given you, in that folder, the BCF file that was called bugsnips, bugsnips.bcf. So that's a binary call for, so variant call format file, but it's compressed into binary. So we need to turn that into something that has genotype approximations in it. So we need that file. We need a file with our phenotype scores in it, which can be continuous or it can be binary. In this case, we're using a binary one. And we need a relatedness matrix. GEMMA can generate the relatedness matrix for us, and we're going to do that right now. So, we don't actually need to make a GWAS directory. We've basically just gone straight to this bit, okay? Straight from our fast data thing. So that's the line of code that you should have run. And if you list what's in that directory, you should have these 2 files and a copy of the PDF, but the PDF you don't actually need. Does anybody not have those 2 files? Put your hand up if you have no idea what I'm talking about. Okay, so everybody has those 2 files, and they're in the same folder. And that folder, you can call it whatever you want. If you've called it GEMMA example, it's going to be a lot easier. If you have those 2 files, we're ready to go. You don't have those 2 files. You don't need to do the make directory GWAS. Okay? You can if you want to, but you should have already just copied those files across so we could save a couple of minutes. Put your hand up if you need help. Put your hand up if you'd like to see the giraffe cartoon again. I don't know how to make that happen. You can download the PDF yourself though. So change directory into your folder called GEMMA example. You should have something like this. You should be seeing this prompt that says GEMMA example. Is everybody there and they have those files in that folder? Yeah? Okay. Let's continue. In our phenotype file, we can have a look at the contents of that using the command less. If you type less bug_binary.pheno, or you can just type bug_ and hit tab to autocomplete, you'll see that it's a single list and it has no identifiers, no headers, no row names. It's just the list of phenotypes. And that's the requirement for a phenotype file to run in GEMMA. Those phenotypes are in the same order as the columns in our binary SNP file. But we can't read the binary file. We need to turn that into something useful, which we're about to do. we're going to create a genotype file. So if you just type in BCF2 bbgeno and hit enter, you'll see this. And this is simply asking you for the name of a SNP input file, which is in binary call format. So here the example of how to run it is BCF to bbgeno -I variance BCF, but we don't have a file called variance BCF. Our file is called bugsnip. Okay, so the whole path to the script will be, so let's do that now. So that will be in fast data, MEGP 2014, scripts, RFseq, tools? No. Okay, maybe it's back. Yep. So that's the, oops. That's the path, the complete path. So fast data, MEGP 2014, scripts, tools, BCF to bbgeno.pl. And then you can use -I for our input file, which conveniently we can still see hanging out just underneath here. I did this on purpose. And we know it's called bugsnip. Bugsnips.bcf. And the default of this program, if you don't specify anything else, it will create a file with the same base name, so it will be bugsnips, but it will be a bbgeno file, which is exactly what we want. And hit return, and it will count up through all the SNPs. And I think there's about 55,000 SNPs. You're out in the. Make sure you're not in. If your command prompt says that you're in login 1, that means you're actually in the head node of Iceberg. So you need to type QRSH and move away from the head node to actually access. And then you need to change directory back into.
Okay,
Yeah. So somebody else will help you. Oh, no, you don't need any more SSHs, no. No, you're in. You can just CD to, like, so CD space / fast data. Okay, so we can have a look at what's in our bbgeno file that we just created. Using less, I'm going to use the -S to stop any soft wrapping of lines, so we just see the beginning of lines. And then the name of our file. So you can see there's no headers in this file, but we don't need any headers. Instead, what we have is the identifier for each SNP, which in this case includes its scaffold and its position in the scaffold, the identity of the major allele, the identity of the minor allele, and the genotype frequency, the genotype estimation for each allele in each sample. So in this case, we had 96 samples, so there'd be 96 columns of numbers. What do these numbers mean, right? That's the question, yeah? Okay, so the minor allele, so the major allele is the allele that's present in the reference sample. And then you have your alternate allele. So the million-dollar question is what's going on with the genotype. So we convert it into a number. If we had absolute certainty in our genotype, we would call it a 0 being homozygote for the reference allele. It would be exactly 1 for a heterozygote and 2 for homozygote for the alternate allele. But because we estimate through a Bayesian process, we only have an estimate. So we can have some uncertainty. We can have 0.1, so it's likely to be a homozygote, but we're not saying it is. And a lot of our pipelines allow for this uncertainty in our genotype calling, which allows us to get away with a little less information and just take into account that we're not certain.
And how many possible alleles are there?
So we restrict it to only doing analyses on biallelic systems. I mean, clearly there could be 3 alleles or 4 alleles or 5 alleles, but it just gets intractable. So we just say, let's just only take SNPs where we have only one alternative.
Okay.
You can certainly do more, but the maths becomes much more complicated. So, did everybody successfully run that script? So now we need to run another script. And what I would recommend you do is use the up arrow to get back to the scripts thing. So you've got that whole path in there already. And then just take off the front bit. I'll show you what I mean. But this is the script that we want to run, is this GBS LMMPL. It's not in the tools folder, it's in the RFSeq pipeline. So it's in scripts, RF, autocomplete. Where did you put it, Victor? Oh, okay. Scripts GWAS. It is in scripts GWAS. And if you just press enter on that and don't actually put any variables in, it will give you kind of an error, and that error is that it just shows you the correct usage. And it tells you all of the things that you could put in, but in reality most of these defaults are set to what you would want, so you don't actually need to enter all of these variables. The only thing that is absolutely required is that you have a geno file, right? Which in this case we have a BB geno file, but it will work just as well. So we want to run that script, and we want to tell it our input file is bugsnip BB geno. Huh? And then we want to tell it that we want to use a Bayesian sparse linear mixed model, BSLMM, and we want to take option 1. So we can see in the usage here that in the BSLMM call, there's 3 different options. We can just run a linear model, but still running in a Bayesian framework, so it's still going to run a Markov chain. We could do a ridge regression, or we could do a probit model. A probit model will only work with binary data, which in this case we have, but it takes quite some time. So for time reasons, we're going to run it just with a linear model. So it's still going to estimate a population of SNPs of no effect, and then a linear model for the rest. Okay? So we want to constrain this to option 1. If you're actually running this on a normal data set, you would probably, on a binary data set, you wouldn't even have to put this in, because the default is to run on the probit model, the way I've set it up. And then I'm also going to restrict this to just running 5 replicate chains, so that we can get some parameters, some better estimates of our parameters. The default here would be to run 10 chains, which you can do. If you want to run 10, run 10. Won't make any difference. Iceberg can handle it. I'm just going to limit it to 5 because it's easier. So if you set that running, what you've actually done is run several commands all at once in this wrapper. And I'll step you through them. But what I'm going to do first is I'm actually going to submit this job, and you guys can do the same. Submit the job, and then that can be running while I'm explaining what this has been doing. Okay, so my array job is running. Okay, so does everybody have that running? Yeah? Okay, so there's the command line. This is the abbreviated command line. So I don't have the fast data scripts, tools, stuff. So that's basically what we've just run. And we've set that running. What this has actually done is first it's pulled out some information. It's just done some awk manipulation, and it's pulled out that first column of the genotype file from the BB geno file, and it's split that into 2, and it's generated a location list of where the SNPs actually are that it can refer back to. So it's done some funky coding on the inside that we didn't have to worry about. That's the first thing that it did. The second thing that it did was create a relatedness matrix of how those individuals and the phenotypes are related to each other. And in this case, it's actually run this command, and it's created a centered relatedness matrix. So we didn't have to worry about that. So we've run one simple command, and it's actually done 3 or 4 steps for us. And if you download the GEMMA manual, it will say the first thing you need to do is generate a relatedness matrix. Here's how to do it. Run this command. So I've just taken it and made everything automated, and I think you guys will get access to this script afterwards. Is that correct, Victor? No, you weren't listening? Okay, it doesn't matter. Okay, the next thing it did was actually to write a script that would run this command. Because I told it 5, it runs this 5 times. And if you told it 10, it would run it 10 times. Or if you wanted to run 100 chains, you could run 100 chains. And then we actually ran it. So what did it pull out? So this CXX, this is the relatedness matrix that we just generated half a second before. And it automatically created a folder called output. And you can list the output using this command, and you should see there'll be a whole bunch of files, and it should have finished by now, I think. Yep, mine's finished. Everybody else is finished? So it's generated a whole bunch of files. It's generated the hyperparameters, which is an estimate of how many, like what the proportion of the entire population of SNPs that go into the SNPs with no effect, the random effects, and the SNPs that have an association with the phenotype. It's generated some estimates for those parameters. It actually gives us those parameters, and I can't remember what's in BV. But now we're going to run another script that will summarize that information for us. And this one it's in the same folder as the GBS LMM one, except it's called GEMMA Summary. Who's completely lost? Nobody lost? Who just doesn't care anymore? Who regrets staying up till 3 a.m. drinking whiskey?
So, can you explain again exactly what we're doing? Because I get the line, but what
Yeah. And then we ran a model. And in that model, we asked, what's the association between the phenotype and each SNP? And we went through individual. And I'm going to show you the summary output in a second, and I'll explain what the numbers mean. Hopefully.
So if you don't have access to any scripts that are for a specific server, but just run GWAS as it is,
Yep.
then all of these things can be done.
You have to do each one individually. Yeah.
But they can be done. I mean, the output we get is a standard output of GWAS.
Yes. Yeah. So, well, each one of these, so all the ones with 5, so Gamma would output these 3 files, sorry, these 4 files, the parameters, the hyperparameters, gamma and BV. Okay? So BV is estimates of the slope, gamma is estimate of the effect size, the hyperparameters is the proportion of SNPs that go into each population, and that's all. And the parameters are actually the parameter estimates that come out of the model. But we need to summarize them so we can make some sense of it. And if we run this next one called Gamma Summary, it will tell you nicely if it finds the correct files. And if it does, it will actually start running the analysis. What is it actually doing? I tried to make this as fast as I could so we could have stuff running, and I'll try and explain it while stuff is running. So we run that command. First thing that script does is it goes and it gathers a whole bunch of information about the files in the output folder. So here we're telling it to look in the for a folder called output, which is the default folder that Gamma always creates. And we tell it to get some other information, and then it actually calls R from inside the script. And you guys have a copy of this PDF now, so you can actually read for yourselves exactly what this script is doing, but it's basically just doing a whole bunch of matrix assimilation and saying, okay, so we had all of these estimates, let's combine these 5 together, what's the mean, and can we calculate a 95% credible interval around all of those mean parameters? Yeah? So here this snippet here is showing you how it's calculating the 95% credible interval for the hyperparameters, which is the proportion of SNPs that go into each population. Everyone's looking either lost or disinterested. Yeah? Okay, let's just move on to the files that it automatically writes out. This is actually the bits that you're interested in. Okay? So we create a summary table, which we'll have a look at in just a second. We create files at different levels of the SNPs that are involved in, the top 1% of SNPs, the top 0.01% of SNPs, the top 0.001 SNPs, which are like the smaller that number, the more likely is they are the SNPs of large effect. Right? Then we have our PIP scores, our posterior inclusion probability. So where we ran all of these models through our chains, can we find a probability of including that same SNP in 25% of models, or 50% of models, or 10% of models, or 1% of models? And basically, if we're not always pulling out the same SNP, it's probably not real. Okay. So let's have a look at some of these outputs. So here's a summary table. This is probably the first thing you want to look at. So this H is the hyperparameter of the proportion of SNPs that have no effect. So the way this works is the first iteration of the model, it says, how many SNPs do you have? So you have 1,000 SNPs. It says, okay, 999 of those SNPs have no effect. If I run a model that says there is one SNP that has an effect, how does that work out? And then if it finds that it can't find a good model, it'll say, okay, so maybe that's no good. Let's try it with 2 SNPs have a large effect and everything else does nothing. And then it will try with 3 SNPs, 4 SNPs, 5 SNPs, etc. And it will cycle through. And in the end, it will stabilize on what it thinks is the right proportion of SNPs that have no effect. And in this case, it's saying that 97% of all of our SNPs have zero effect. And then the next two columns are actually the upper and lower equal probability tail intervals. Okay, so that's just saying what's our 95% credible interval around this estimate. And so in this case, this is saying between 88% and 99% of SNPs have zero association with our phenotype. Now, I find that difficult to believe. That means that whatever SNPs we found explain all of the variants that we can explain. And the variants we can explain with our SNPs that have large effect and our random effects, our SNPs with no effect, is this value here, the proportion of variance explained, PVE. So this is saying we can explain 97.5% of the variance in our phenotype using these models and these parameters that we've pulled out. Who thinks this might be real? Put your hand up if you think we found, before we get to the end of the summary table, the answer. Put your hand up if you're a monkey. So our credible interval here, again, is 90 to 99%. So it's saying we're explaining nearly all the variation in our phenotype using, and our hyperparameter says it's a very small number of SNPs that can explain all of that. And if we look just at what that small number of SNPs can explain, it can explain 93% of this 97%. That's basically saying we have found the SNP that controls this phenotype. And then this last line tells us how many SNPs we needed to do that. And this is saying we need 21 SNPs. Using 21 SNPs, we can explain this phenotype with an accuracy of 97%.
21.8?
Yeah, so 22 SNPs. Well, it's Bayesian, right? So we're not quite sure. We could say, okay, 22, but it's somewhere between 13 and 28. So somewhere between 13 and 28.2. I could actually change this table so it rounds to the nearest whole number so that it's a little bit easier conceptually. But the reason I did this is because this is saying we can explain 98%. Now, unless you're dealing with a single SNP of a massive effect, like sickle cell anemia, I think this is wrong. And I would caution always, if you can explain more than 60% in total, you've either done a very tightly controlled cell culture experiment, or there might be issues with the way you've run your model. And we're going to come to some of those in a second. If we look at SNPs that have a posterior inclusion probability of greater than 25%, we have a whole bunch of SNPs here. And actually, because I know what the true answer is for this data set, or for the whole data set, this one here, on linkage group 8, is probably real. All the others, almost certainly not. But we get a list, and we get this second last column is the posterior inclusion probability. Look at this. We have two SNPs that in 100% of models, they're always there. That's saying, that this SNP is real. But we have to remember that because our hyperparameters suggested we can explain so much with such few data, it's probably an artifact. If we look at our top 0.1% of SNPs, then we see that we still have 5 SNPs that are likely to be involved. What we can then do is we can go back and extract the sequence around those SNPs, and we can blast that sequence and say, is it in a protein coding region? Does it make sense? You found something that's associated with a black phenotype, and if you find that SNP is in a coding region for melanin, maybe it's real. But if it's not, you would really have to question it. Here I've just plotted the posterior inclusion probability, and I've scaled the point size based on that that posterior inclusion probability, just to highlight where those SNPs of really large effect are. And this red line I've put on here is just at the 50% mark. So if you trusted these data, you would say that, okay, we need to investigate all of these big dots, because it looks like, you know, we have a positive association in at least 2 different places on the genome that we could then start to investigate. If we ran a much more appropriate model, which would be in this case the probit model because it's binary data, we'd get very different results. So now it's saying that actually it's only 3/4 of the SNPs that have no effect, and that 25% of the SNPs do have some kind of an effect. But now we can only explain 45% of that 75%. So now we can explain about 30% of the phenotypic variation using some SNPs. But our estimate of how many SNPs we need is somewhere between 3 SNPs and 140 SNPs. So this is probably much closer to the truth in that we don't have an association with our phenotype with these data.
What did we exactly change?
So this we ran a probit model. So a probit model. So instead of running just a linear model, we've looked at before we ran a linear model. So before we ran on this association, all of our data was up here and down here because we only had 0s and 1s. But then when we run a probit model, that becomes a probability distribution with all of our 1s up here and all of our 0s down here. And we ignore all of that, and we just take this slope, and that's the probit section of the regression. And so we assign to each individual a probability, and we get a much better estimate. And so if you have very skewed samples or very low numbers of individuals, it's really important to run the right model. In fact, it's always important to run the right model. But particularly with GWAS, they're very biased to only finding SNPs of very large effect. Everybody wants to die. So I think that's actually the last of my slides. I have one more. What does it say? Okay. So this is actually.
How do you know which will be the good model to run?
If you have binary data, you should probably always run the probit model. If you have continuous data, you can't run a probit model. You can't. You cannot.
So you cannot run which?
You cannot run a probit model with continuous data. But what I would recommend is always to, if you have continuous data, I would run the model and see if you get a good estimate, and then convert that data into binary data. So make your arbitrary cutoff and say, okay, if it was over this value, it gets a 1. If it was under, it gets a 0. And then run a probit model and see if you get the same SNPs coming out. If the association is real, pretty much whatever you do, you will get the association. But the real big message is make sure that it's not getting stuck. If all your parameters are up in the high 90s, it means it's a really, really poor fit of the model. And this is the posterior inclusion probability when we run the correct model for this data. And you can see that there's really no pattern there, and especially if you look at actually what these inclusion probabilities are, it's basically saying every SNP is like less than 2% likely to be included in any one model. So we do not have an association in this trimmed down data set. But then again, we only had 2 million reads spread across 100 individuals, so we just didn't have any power at all.
With the summary table in the linear model, so here we have no association between the phenotype
That's correct.
When you get to the even that one, it's telling you that you have like a 77.
Well, no, it's not, because look at our 95% credible intervals here. We've got between 5 and 90%, and the truth is in there somewhere. And I suspect it's much lower to this, much closer to this end, especially when we look at our posterior inclusion probabilities and see that there's actually no SNP that sticks out. There are other methods that you can use for doing GWAS. You could use GENABLE, which doesn't take into account all SNPs simultaneously, which GEMMA does. So you would then actually look at each individual SNP. Is this SNP associated with this phenotype? And you would get a score for every single SNP. And you'll get much broader regions of genomes might show an association, but you won't actually get any causative SNPs. So I think now we might step back and go back and do some mapping. Are you guys more interested in doing mapping of reads back to our reference genome, or population structure analysis, or do you want to just have a chat about downstream analyses? You guys are the ones who are here to learn. You can
know something about population structure estimate?
yeah.
I'm interested in association.
So more association studies, structure. So we've still got mapping and SNP calling. Everybody confident that they can just do that, or do you want to go through that?
Can you just remind quickly which are the lines here? H, R, H, O, G.
So H is the proportion of SNPs that belong to the population with no effect or with very small effect. Okay. So when we split our population of SNPs into these separate populations, so SNPs with large effect, so if this was effect size, so things that have a big effect are over here in this population of SNPs, things with small effect and things with no effect. This red one, the no effect, that's H. That's the proportion of all SNPs that are in that random effects, the not significantly associated with the phenotype population. Yep, that was the first one. So rho is just another one of those hyperparameters. Rho, I can't really, it's in the GEMMA manual. I think it's the relationship between the variances of the different populations of SNPs. But the big ones are PVE and PGE. Okay? So PVE is the proportion of variance explained. But that's the proportion of variance that's explained by that red population and the black population together. Then PGE is the proportion that's explained just by the black population. So just the SNPs of large effect without the SNPs that are unrelated to the phenotype. You don't look happy.
Can you repeat like the last thing?
So PVE, I'll put it on the board.
PVE is
Yeah.
all
So PVE is equal to the SNPs with association plus the no effect Okay, so including random effects is PVE. PGE is only those. Yeah? Okay, so PVE is both these things. PGE is just that population. Yeah, PVE is the amount of including all SNPs. PGE is only the SNPs with significant association with the phenotype.
But the phenotype is binary, so
Yeah.
it's
So,
not like
yeah.
one have a phenotype and the other have nothing. The other
But it's the so that variance still gets shunted into random effects, right? So in a mixed model, you have covariates, these things, plus error that you're trying to explain, and in this model, all of the SNPs with no effect get put into the error, and so we can partition out just the SNPs of large effect. And then you can calculate the absolute effect size by multiplying for each SNP PVE by PGE. Will give you the effect size for each individual SNP. So it would be in this case, this is the bad one, remember, but this would be 93 by 97. So what's that? 93% of 97 is 90%, más o menos. And normally, if that number is somewhere in the 40% range, it's probably getting close to reality. And in those other parameter files, the gamma and the BV files, you can actually get the estimates for individual SNPs, and they're listed in exactly the same order as the SNP location file that you generate at the same time. So if you're there are many other models that you can run. This is just using the Bayesian models that I've been talking about. You just need to use your common sense. You know? If you actually think that your phenotype is that strong that you can, that 97% seems reasonable, the first thing you would do is to identify which SNPs have really high posterior inclusion probability. Go back to your genotype file, extract that consensus sequence, and blast it, and see if it's coming out in a coding region and it's associated with with the phenotype that you can plausibly biologically explain. Yeah, yeah. It would be okay. So now, could this gene actually be associated? And then you would go and look at your RNA-seq data or do further studies. Because you're only ever identifying a correlation. You're not able to show causality unless you go out and actually do an experiment. Like a knockout recovery is pretty much the only way to go. But this is a great way to find candidates for your knockout studies.
Sure, one question. If we want to study to detect SNPs associated to a particular character, for example, a shell coiler, how many individuals do you have?
Okay, so GWAS are really subject to ascertainment bias. The more the better. So way back at the beginning of this.
Can you show us so phenotype files, what it looks like? I'm more used to genotype files.
So this is the phenotype file basically. Okay. It's just a single column of the phenotype. And in this case, it was a binary phenotype, presence or absence of a character. And it doesn't matter what that character was. In this case, it was being melanistic morph to MEMA. But this could be I don't know, being an alate aphid, like having wings or not, or it could be being male versus female, or it's just a toy data set.
Can you have an intermediate phenotype? Like here
Yes.
you have only
Yeah.
0 and 1.
Yeah.
You have like several other.
Yeah, yeah. You can have absolutely. You can have continuous phenotypes. You can have a categorical phenotype, scored as 0s, 1s, 2s, 3s, 4s. But you can only run a probit model with binary presence absence or 1-0 data. Apart from that, you would have to run the linear model or a ridge regression. Or you can forget the Bayesian approach and you can actually do a whole multivariate approach, and you can, have a 4-column phenotype file and submit that as the job. And it will be very fast, but because it's not a Bayesian approach, you're going to have a more solid estimate. You're not going to have any hyperparameters, so you're not going to split it into multiple populations. It's going to assume it's a single population of SNPs with an underlying distribution, and that may not be the case because there could be some SNPs that just have absolutely no association, which is why we like the Bayesian approach. Your question? Thousands. The more, the better. So here, these population sizes here. So these are SNPs with 5% effect. So this is the strength of the effect: 5%, 10%, 20%, and these are the sample sizes and the detection probabilities. And you can see as the sample size goes up, your detection probability increases. For most human disease stuff, tens of thousands of samples. But when you're assigning genotypes, that's based on the whole population. So the larger the population, the more certain you are about your genotype.